we are going to discuss on reverse transcriptase PCR. At first, here the term is mentioned that reverse transcriptase. In my last class, I was discussed on the PCR, but in that class, the material for the PCR was DNA. Here it is mentioned that the PCR is associated with the reverse transcription. Before discussing on the reverse transcription, it is necessary for us to know about the nature of different types of polymerases associated with the nucleic acid. Basically, the polymerase enzymes are responsible for formation of 3-5- polymerization of phosphodiester. Again, I am writing here that it is the ribose sugar or deoxyribose sugar, whatsoever may be. And here it is the nitrogenous base, and here it is the phosphoric acid. It represents a basic organizational pattern of nucleotide. Again, the other nucleotide is like this. It is the nitrogenous base and it is the phosphoric acid. Here again a nucleotide, other nucleotide, nitrogenous base and phosphoric acid. Actually, the mononucleotides here, all are the mononucleotides, and we are referring here the polymerase. We are referring here polymerase. The name clearly represents that the enzyme is responsible for the polymerization, or in other way, it is better to say that the enzyme is responsible for the formation of a bond between two structural units. It is the one structural unit, it is another structural unit, and here the third point of the first nucleotide and the fifth point of the fifth position of carbon of the second nucleotide are involved in the formation of phosphoester bond. Again here the acid, the ester bond, here the second phosphoester bond. That means it is responsible for 3-5- phosphoester bond. It is responsible for the formation of the 3-5- phosphoester bond. And now if you consider the enzyme polymerase, that means on the basis of the source, the polymerases are observe that DNA dependent DNA polymerase. DNA dependent DNA polymerase. And that DNA dependent DNA polymerase is responsible for the synthesis DNA strand by the use of the DNA template strand. Second, DNA dependent RNA polymerase is also responsible for the polymerization and formation of the phosphodiester of the ribonucleotide, but by the use of the polyester of the template strand of the DNA. Third, RNA dependent RNA polymerase. RNA dependent DNA polymerase and RNA dependent RNA polymerase. That means it is the basic and fundamental principle which is necessary to explain here and analyze it. Here, the DNA is synthesized, that is by the polymerization of the dideoxyribonucleotide. By the use of the DNA template strand here, and it is responsible for the replication. We all know that the enzyme is responsible for the replication and commonly referred as the DNA polymerase 1, DNA polymerase 2, DNA polymerase 3 in the prokaryotic cells, and DNA polymerase alpha, beta, gamma, delta, and beta in the eukaryotic cells. And actually, it is responsible for the what I will say, the replication. The second, DNA dependent RNA polymerase, and a large number of the RNA polymerases are observed and studied in the B.Sc classes that are in the enzymes are responsible for the transcription process, and that is responsible for the synthesis of RNA by the use of the DNA template. Third one is the RNA dependent DNA polymerase. Here, it is important that it is responsible for that enzyme is responsible for the synthesis of the DNA strand by the use of the template of the RNA. And at first, the enzyme was reported by Baltimore and Temin, reported and explained by the Baltimore and Temin in retroviruses. And the reporting of the RNA dependent RNA polymerase, or it is better known as reverse transcription. It is referred as reverse transcription, and that particular enzyme is used for in the urgent PCR. Why so? It is referred as reverse transcription PCR method. And the reverse transcription is here the reporting of the reverse transcription from the retro group of viruses. Retro group of viruses. The enzyme was at first reported by the retro group of viruses, like AIDS. AIDS is also the retro group virus. Retro group of viruses, and it is responsible for the reverse transcription, and it is responsible for the change of the concept of the central dogma presented by the Crick, and that is the flow of the genetic message. Fourth one is the RNA dependent RNA polymerase. Fourth one is the RNA dependent RNA polymerase. That means the RNA is synthesized by the use of the RNA template, and in better to write here RDRP, and RDRP is most recently observed in the coronavirus. RDRP is observed in the coronaviruses nowadays reported, and which is burning topic nowadays. Here, by the use of the reverse transcription, we can able to synthesize DNA template from the RNA. Now we move further to see. Here, the reverse transcription, or it is a simple technique. Before the development of the concept of the reverse transcription, and by the use of the reverse transcription, we are in a position to develop the desired number of the DNA sequence for the study of DNA sequencing or the analysis of DNA expression of gene and a study of the virological genomic sequences. And there are a lot of the medical and forensic techniques which are based on the reverse transcription. Before the development of the reverse transcription, the method which was used, or nowadays at present it is also used in some laboratories for the identification and detection of the RNA, particularly the mRNA, is northern blotting technique. But the northern blotting technique is superseded nowadays by the reverse transcription PCR technique. And here the reverse transcription technique, in one way reverse transcription technique is responsible for the detection of the RNA, and in another way it is also responsible for the analysis of quantitative value of the RNA and provide help in the identification and detection of the viral infections and intensity of the viral infections during the treatment. Hence, the PCR, sometimes RT-PCR is referred by the real-time PCR, but some here differences here. The reverse transcriptase PCR and second one is qPCR, quantitative PCR. It is in short referred as RT-PCR and another qPCR, that is quantitative PCR. The RT-PCR is useful in the study of molecular cloning, sequencing, simple detection of the RNA, etc. And qPCR provides help in the quantity of the copy of the number specified piece of the DNA. And these are the techniques which I am going to just discuss in present class also. And before coming on the details, it is necessary to at first understand what is the proper principle and method for the sequences, obtaining of the sequences from the mRNA by reverse transcripted PCR. Here we know all that it is the mRNA. It is the mRNA, and mRNA, particularly in the eukaryote and in some viruses also, like in coronavirus, in some viruses also, in mRNA, 5' end has AUG initiation codon and 3' end is poly T. These are the characteristics of the mRNA. And 5' here 3', at the 3' end, mRNA has a polymerized tail, which is known as the poly T, and at the 5' end, a 7-guanidinic cap is present. That is referred as 7-guanidinic cap. Guanidinic cap. But here we have no need of the 7-guanidinic cap, because the 7-guanidinic cap is associated with the ribosomal attachment and help in the protein synthesis. Here the sequence, we have no need of this sequence, but here we have the poly T. In the last slide, or in my last representation, I was discussed that during the polymerization, most important thing is the primer, and here we also need primer. And for commonly, of course, the 3 types of primers are used in the reverse transcriptase PCR, which I am going to discuss it. But here, because we have the poly T here, polyadenylate tail present on the third 3' end of the mRNA, we simply use the poly A tail. A. Then we use the primer of the poly T. Poly T. We use the poly T, and here we provide the temperature at the from 37 degrees centigrade to 4 degrees centigrade, and when we light this 4 degrees centigrade, we provide and here during the 5'. Now the primer is attached, and after the attachment, it provides a base for the attachment of enzyme reverse transcriptase. And just we are discussing that the enzyme reverse transcriptase is responsible for the synthesis of DNA template from the RNA. That means the reverse transcriptase is RNA dependent DNA polymerase, and here it is the polymerase which is responsible for polymerization of the strand. In this way, we observe here that is hybrid of the RNA DNA is formed. But here the DNA is complementary to the mRNA or RNA used, and it is single. Here the reverse transcriptase. We use the reverse transcriptase, and after the polymerization, commonly sometimes the strands are separated in some technique, in some processes. The strands are separated, and which is responsible for the formation of a single strand mRNA or a single strand DNA, or the DNA is secondarily polymerized by the DNA polymer to form the double-stranded DNA. But sometimes the DNA, the polymerase, has the RNase activity. The polymerase has the RNase activity, or if the RNase activity is not present, then we add RNase H. That RNase H is responsible for the lysis or breaking of the RNA. And that is responsible for the formation of. Now the lysis is used, and we use DNA polymerase T4 DNA this part and responsible for synthesis of this strain. And in this way, a double-stranded DNA is synthesized from the single-stranded complementary DNA, and the RNA sequence is liked by RNH, where the component associated, a gap is available here, that is filled by, niche is filled by enzyme ligase. In this way, we obtained a double-stranded DNA from messenger RNA, and the sequences are same as we initiated from the messenger RNA. It is the basic principle of the synthesizing of our DNA in reverse transcriptase polymerase chain reaction. Two things are important here. One is reverse transcriptase and another is primer. The reverse transcriptase we obtained from the two hormones mentioned here, MMLV reverse transcriptase and AMV reverse transcriptase. MMLV reverse transcriptase, MML virus reverse transcriptase and AM virus reverse transcriptase. These two reverse transcriptase are used for the, either one are used for the reverse transcription or reverse synthesis of the DNA from RNA. Next, the second one is the primer. Here we go. Here we observe the primer. In the primer, commonly the three primers. One, oligo poly T primer. Second, sequence specific primer, and third, random primer. These three forms of the primers are used. We use these three forms of the primers, and the selection of the primers depend on the pattern and use of the assay. We just what we want to analyze, it depends on that particular process. That which one is selected, that is oligo poly T, oligomerization of the thymidinic ribonucleotides, sequence specific primer, when we want to analyze some particular sequence, and then we use the primer a specific primer, and generally, if we use the all RNAs without any separations, we collectively the tRNA, mRNA, rRNA collectively, then we commonly use the random primers, which we will discuss here. And after the polymerization, we use the primer, and then the second the complemented DNA, which we obtained by the process of the reverse transcriptase or reverse transcription. Complementary DNA. Then the complementary DNA, from the one complementary DNA, we use the same PCR, that is common PCR, that is denaturation or NELE, and elongation as commonly PCR, and it includes the one cycle. And now we repeat the cycle, then we get the total number of DNA as 2 power n. In this way, we collected the large number of DNA of the desired sequence of the messenger RNA or the viral genetic material which we want to describe or analyze, and the complete process is referred as reverse transcriptase PCR. And when we use the reverse transcriptase PCR, in the process, the reverse transcriptase PCR, one is the primer, second is the reverse transcriptase, third is the ligase. Ligase is commonly collected from the T4 bacteriophage, and the T7 bacteriophage DNA polymerase, which is extracted and used in the reverse transcriptase PCR, and it is associated with the replication and DNA synthesis of the T7 bacteriophage, which has potency to synthesize double strand of the DNA, which is most specific feature of the T7 polymerase enzyme. And the process, if we observe the process, the reverse transcriptase PCR of the two types, one step RT-PCR and double step RT-PCR. If you observe the process, it is simply the one step PCR in one step PCR we use RNA primer, and here the primer is sequence specific. In this condition, the primer is sequence specific. Enzyme reverse transcriptase, polymerase, deoxyribo ATP, deoxyribo TTP, deoxyribo CTP, deoxyribo GTP, plus buffers, required buffers, plus ions plus and all are placed in one step depend of or tube now we get in one step the complementary DNA, then we get the amplified form of the DNA from the RNA. It is referred as the one step reverse transcriptase PCR. In the two step reverse transcriptase PCR, in first step we use RNA. Here non-specific primer plus reverse transcriptase D ATP D TTP D CTP D GTP buffers and ions, buffer plus ions and reagents. And here it is responsible for, it is in the one step, and in first step it is responsible for the reverse transcription, and it complementary DNA. It is responsible for the formation of complementary DNA. Now after the complementary DNA, we use the specific primer plus polymerase plus other reagents required for the PCR reagents, and now it is responsible for the synthesis of large number of the DNA which we require for the study. In this process, there are two processes, and each process has some benefits and some drawbacks. And now a very simple machines and kits are available in the market by which we can easily able to detect the RNA and polymerize the DNA from in the laboratory. Now, it is the reverse transcriptase PCR, and the reverse transcriptase PCR is secondarily used for the quantitative PCR, RT-PCR, and it is better to say that RT-qPCR. RT-qPCR. That means the quantitative PCR, the process of the quantitative PCR is always followed by the RT-PCR. qPCR is individually the qPCR is not possible. Then we use the RT-PCR, and then the qPCR is followed. In the qPCR, the RNA transcripts are quantified by reverse transcribing them into complementary DNA first, and then qPCR is subsequently analyzed. In the qPCR, name clearly suggests that the qPCR is responsible for the quantitative analysis of the RNA in any sample or in any cell or in any medium. And the qPCR again, the qPCR is of two types. In one process, we use the fluorescent dye-based qPCR, which is commonly known as dye-based qPCR. Dye-based qPCR, and second is the probe-based qPCR. One is the dye-based qPCR, and another is the probe-based qPCR. In the dye-based qPCR, commonly use the fluorescent dye, and the fluorescence is commonly measured. The intensity of light emitted by the complementary DNA amplified in the medium is measured, and that the intensity of light is responsible for the quantification and analysis of the quantity of the RNA present in the medium or in the cell. In the probe-based qPCR, the most commonly used probe is the hydrolyzing probe, and when we use the hydrolyzing probe, here the one is the fluorophase, and another is the quencher and fluoro. The quencher and fluoro base is responsible for the helpful in the FRET, fluorescence resonance energy transference, and that the interference level by the probe when it is probe is dissociated from the complementary DNA during the polymerization, it quantifies the measurement, and that the intensity is gradually increased, which is responsible for the analysis, and it provides a tools of measurement for the quantity of the RNA of the. Hence, here the reverse transcriptase PCR or qPCR, and it is better to say that the quantification is the determined by a time by which all the mRNA is converted into the DNA. So it is also referred as a real-time PCR. In this way, the reverse transcriptase PCR provides help in the analysis of gene expression, virological study, genetic analysis, DNA sequencing, and proteolytic configurational analysis. Because in eukaryotic cell, if you observe for example, I am just saying for example, that in the eukaryotic genetic material, we have intronic sequences and exonic sequences. The intronic sequences are lysed, and the intronic sequences are non-translating sequence, whereas the exonic sequence is translating sequence. And that exonic sequence is particularly involved in the protein synthesis, and hence it is responsible for the determination of the structure and configuration of the. So, if we want to study the complete sequence of the DNA, then it is very difficult to analyze the specific sequence which is responsible for the formation of the particular polypeptide chain. And here, if we study the sequence of the mRNA converted into a DNA, and then we match that synthesized complementary DNA with the original genome, then we can easily analyze that which part is converted into the exonic sequence and which part are still remain in the non-translated form. In this way, we can easily analyze the other regulatory sequences of the DNA, particularly in eukaryotic cells. In third way, nowadays in virological study, if we want to study the RNA, quantification of the RNA, degree of infection, like this, at present in the coronavirus. And it is very interesting here, just I am telling you, that in the coronavirus, the mRNA is, corona is the RNA containing viruses. Commonly, corona has 2.8 to 3.1 kilobase nucleotides, and the most important thing is that the RNA of the corona group is resembled with the eukaryotic mRNA. Why? Because the DNA, the RNA of the corona group contains 5' as the mRNA of the eukaryotic cell, and 3' polytail. Both are characteristically observed in the eukaryotic genetic material, and due to the presence of these two characters, the RNA, it is the positive strand. The RNA easily entered, when entered in the host cell, it starts the protein synthesis. That means it is in the corona group, it is responsible for the transcription and replication, both replication of RNA and transcription, because just I told you in this class that it has the RNA dependent RNA polymerase enzyme that is responsible for the synthesis of the RNA, and the presence of this form is responsible for the just as the protein synthesis tools, and it is the one of the basic cause. They are all the cause. It is one of the basic cause for the rapid expression and spreading of the corona group. Thank you. I think that the class is useful for you. Thank you. Thank you very much.